Okay, class, so here's part two, and we're going to be doing stem cells. Two important terms you need to know whenever you're talking about stem cells is the concept of pluripotent stem cells and multipotent stem cells. So, multipotent stem cells can differentiate into almost any uh, cell type. So you can almost think that, so a fertilized egg, for example, is a pluripotent stem cell. A multipotent stem cell has all the characteristics of a stem cell, but it's limited in the number of cells that it can differentiate into. And that's the key difference between them. So embryonic stem cells, they're pluripotent, whereas hematopoietic stem cells, the stem cells that give rise to all of the cells in our bloodstream, are considered multipotent. And then there's a special class of stem cells which came out about a decade ago, and these are called iPS cells. And they're induced pluripotent stem cells. And they're actually made from fibroblast cells from any animal. So you can take like a human skin graft, get the fibroblasts out, and then convert them into iPS cells, and then they can be differentiated into any cell type. And I'll explain that at the end of the lecture. So we covered this when we were talking about cancer, and so you can have a stem cell, and then it divides, and then one of the things that happens when it divides, it self-renews. Okay, so the stem cell actually replaces itself, and it can also differentiate. So those are the two characteristics of a stem cell. They can actually divide and replenish themselves with self-renewal, or they can, or the cells that divide can basically become a transit amplifying cell, and then it'll eventually become terminally differentiated into whatever cell type it was programmed to become. And so here's where the transit amplifying cells come in. So you have your stem cell undergoing self-renewal, and meanwhile one of the cells can actually become a committed transit amplifying cells. So this will basically mean that it goes to become a certain cell type, and then that can actually amplify into many more copies, and then it can eventually become a lot of terminally differentiated cells. Okay, so what ends up happening is you basically have one stem cell that gives rise to many different terminally differentiated cells. And if you guys just go back to the cancer lecture, we talked about this when we were talking about cancer stem cells, okay, which is one of the things that makes cancer such a difficult thing to treat. So where this all comes into play in one area is basically in the lumen of your gut. Okay, so here is just sort of your intestinal epithelium. And these are the villi, and then you have your absorptive cells that are in blue and your secretory cells which are in red. And down here in gray is actually the stem cells, and they arise from the crypt, and as they move out they start to develop. And so down here the Wnt pathway is active, leading to cell proliferation, and then once you get terminally differentiated, the Wnt pathway becomes inactive and there's no cell proliferation. And so what you have down in the crypt are stem cells, and these will undergo division and self-renewal, and then you get your transit amplifying cells, or your that can eventually become like a secretory cell progenitor. And when these are starting to differentiate, they have lateral inhibition. So each one is sort of telling the other one, I want to be this cell, you shouldn't be, and vice versa. And so you have the other one that is basically developing into an absorptive cell, and that has Notch activated, the Notch signaling pathway. And so Notch is inactive over here. And so eventually these two things, there's some crosstalk and some negative feedback between the two cells, till eventually you get development of the absorptive cells, and then the secretory cells in sort of the in your gut here. So this would be equivalent to your small intestine. Okay. So here's some of those cell types that I was just talking about. So here's just an image of an absorptive cell. You can see the microvilli up here, and they're basically going to take in nutrients. from the small intestine. We've seen the goblet cell before, and that's a secretory cell, so all of these vesicles are going to go out and help with digestion and secretion and uptake. And then you have an enteroendocrine cell, which is releasing hormones, and then a panth cell, which is also a secreting cell. So these are just some of the types of the intestinal epithelial cells that are there, that are basically developing from these stem cells right here. Now we see the same sort of thing in hematopoiesis, and hematopoiesis happens in the bone marrow basically, and you have a multipotent hematopoietic stem cell to start that can undergo self-renewal. Okay, this is not a pluripotent stem cell, it's a multipotent stem cell. And then that'll develop into a multipotent hematopoietic progenitor cell, which then it can decide whether or not it wants to be a common lymphoid progenitor or a common myeloid progenitor. If it's a lymphoid progenitor, it'll eventually give rise to natural killer cells, T cells, B cells, and dendritic cells. Whereas if it's a common myeloid progenitor, you'll get macrophages, neutrophils, eosinophils, basophils, mast cells, and the megakaryocytes will eventually form the platelets, and then your erythrocyte. Okay, so your red blood cell. So here's basically how all of the cells develop. This is a good thing for you guys to know, just because it's such an area of amazing research on how all these different things develop. And a lot of times you can actually do this in the laboratory. You can convert these to cells from getting bone marrow. So it has a lot to do with, if you have leukemia and you need a bone marrow transplant, this is why, because the whatever cell you need to be replenished with good cells, and so you can just get a bone marrow transplant. Now, what keeps stem cells, or gives them their stemness, is basically stromal cells. Okay, so you'll have a stem cell associated with a stromal cell, and then you have KIT and KIT ligand, and this basically tells the cell when it's associated with the stromal cell that it should be a stem cell. When it divides and it no longer has that sort of KIT interaction with the KIT ligand, it becomes a transit amplifying cell, or basically, and it goes on to differentiate, or it dies. Okay, so in general, you need sort of this connection and this crosstalk to basically keep the cell a stem cell. In the case with red blood cells, they actually lose their nucleus. So the erythroblasts shown over here will eventually expel their nucleus, and then they become a reticulocyte, or a red blood cell. Now, what's kind of cool is that you can actually take stem cells from a bunch of different sources, and you can differentiate them into various cell types. And so scientists have sort of figured out what to add to the media to get cells to differentiate into a specific cell type. And this has a real lot of promise in an enormous number of biomedical applications. And so sort of think if you had spinal cord injury, right? If you could add some, or any kind of tissue injury, and you needed to repair it, if you could get stem cells, and so in this case they're using embryonic stem cells, and they're culturing them, and then they can add just certain reagents like retinoic acid, insulin, thyroid hormone. You can buy these basically all over the place, and then that develops into an adipocyte. Or if you just add retinoic acid, it can differentiate into a neuron. Macrophages will require macrophage colony stimulating factor and IL-3 and IL-1, and you can add other things and convert them into smooth muscle cells, or oligodendrocytes or astrocytes. And basically they have this worked out for a whole bunch of different cell types. So what's kind of neat here is that you can actually take these embryonic stem cells and you can culture them. And as a matter of fact, this is sort of an interesting story. My cousin, who has a couple of teenage girls, when she gave birth to the girls, they actually took some of her stem cells from the placenta and they basically froze them down for her with the idea, and this happened like 13, 14 years ago, with the idea that if she ever got sick, they could probably at that point in time be able to differentiate those stem cells into a tissue or an organ that she needs. So it's just kind of a cool little thing to add to this. And they're actually pursuing that in a lot of different areas right now in science. And one of the ways they're really progressing with this is they can actually make IPS cells, or induced pluripotent stem cells. So they can take adult fibroblast cells, add these IPS reprogramming factors, which include the transcription factors SOX2, OCT4, KLF4, MYC, and NANOG, and then they throw in a little microRNA called Lin28, and that converts the cells into IPS cells. And then depending on what you add to those IPS cells, you can generate cardiac muscle, skeletal muscle, and so you can see how this could be very useful if someone had a heart attack, for example. They could just take some of their skin cells, differentiate them into cardiac muscle cells, and hopefully regenerate some of the dead heart tissue that occurred because of the heart attack. And spinal injuries are another one. I actually saw a news report a couple years ago, this was probably 4 or 5 years ago, where someone needed to have their trachea replaced. And so what they did was they basically took a piece of plastic, coated it with some collagen and other things like that, and basically took embryonic stem cells and put them in and grew them on the trachea so that they could actually have a tracheal graft put in for the patient who had like cancer of the trachea or something like that. I forget what it exactly was, but I think you guys can really sort of see the promise of this, right? Any kind of injury or anything to the body, these stem cells actually have a very useful purpose. One other side story, because this is sort of the last slide, but it's sort of interesting. My cousin had to have a knee replacement, and she was actually in the same hospital at the same time when Kobe Bryant was in the hospital getting some stuff done for his ankle, which he injured playing basketball back when he was a Laker. And one of the treatments, and the reason they were both in the same hospital and both basically in the same ward of the same hospital, was that they were doing stem cell treatments to their cartilage and in and around where the injuries had occurred to help it regenerate faster. And so it was a novel treatment back then. That happened probably about 10 years ago. So I guess the point is that, being able to reprogram stem cells is just one of these sort of medical advancements that's here to stay, and it's only going to grow, and it's just a really sort of an amazing technology. So those of you guys that are interested, I encourage you to seek out more information from people who do a lot of work with stem cells. Okay, so that's it for